Thermogravimetric Analysis, or TGA, is a powerful analytical technique used in pharmaceutical development to understand thermal stability and decomposition behavior. In this presentation, we will explore how TGA works, what information it provides, and why it plays a critical role in ensuring the quality, stability, and performance of pharmaceutical products. Thermogravimetric analysis measures the change in mass of a material as a function of temperature or time. Typically, a small sample weighing between 5 and 20 milligrams is heated under a controlled atmosphere. As the temperature increases, the instrument continuously records weight changes, allowing us to detect moisture loss, solvent evaporation, and decomposition events with high precision. A TGA curve provides critical insights into material behavior. Initial weight loss often represents moisture loss. Subsequent steps may indicate solvent evaporation. Larger drops in mass reveal excipient degradation or active pharmaceutical ingredient, or API, breakdown. Each step on the curve tells a story about how the material responds to heat. TGA helps answer essential development questions. It reveals whether an API is hygroscopic, whether an excipient can degrade during processing, and whether a formulation remains stable under accelerated conditions. For APIs, TGA determines decomposition onset temperatures and compares polymorphs, salts, and hydrates. For excipients, it evaluates thermal stability and moisture release. For finished drug products, TGA supports stability assessment and regulatory submissions. Think of a TGA curve as a timeline of thermal events. The first small weight loss below 120 degrees Celsius usually indicates surface moisture or bound water. The next step reflects solvent loss, such as residual solvents or granulation water. The major drop in mass marks the onset of decomposition and defines the thermal stability limit of the material. Unlike traditional loss on drying tests, which only show how much weight is lost, TGA tells us when weight loss occurs. This timing information helps optimize drying temperatures, select appropriate granulation methods, and choose packaging materials based on moisture sensitivity. TGA clearly distinguishes between free water, bound water, and residual solvents based on their characteristic temperature ranges. For APIs, decomposition onset temperature is a critical parameter because it defines safe processing limits. High-temperature processes, such as those approaching 180 degrees Celsius, may pose stability risks. TGA also allows comparison of different solid forms, including polymorphs, salts, and hydrates, which may share identical chemistry but display very different thermal behaviors. A common misconception is that if the API is stable, the formulation must also be stable. This is often not true. Excipients can degrade during processing, oxidize over time, or release moisture that triggers API degradation. TGA identifies excipients with low thermal stability or delayed moisture release, which is especially critical in processes like wet granulation, hot melt extrusion, and dry powder inhaler formulation. TGA is a valuable tool for compatibility testing. By analyzing the API alone, the excipient alone, and their physical mixture, interactions can be detected early. In one real-world case, a tablet failed accelerated stability testing with dropping assay values and no clear degradation peaks in HPLC. TGA revealed that an excipient released bound moisture between 80 and 100 degrees Celsius, triggering API hydrolysis. By changing the excipient grade and adjusting drying conditions, the stability issue was resolved and the product successfully launched. Thermogravimetric analysis is more than a thermal test. It is a decision-making tool. It reveals thermal fingerprints, predicts stability failures before they reach the market, and guides processing and formulation choices. When you learn to read a TGA curve like a story, it tells you exactly how your drug behaves under heat. That insight is invaluable in pharmaceutical development. Thank you.